Wavelet distillation from DNNs.
Okay, yeah. So this is a change up here. From the first box, thank you very much.
It's going to be. It's not letting me go. Okay. Okay. So we all agree that image sets have been really important for development of deep learning, especially ImageNet and CFAR and MNIST. You see a lot of that in academic talks. There are actually lots of other very interesting images in science, from cellular biology, cosmology, and also the videos like the protein folding. So the work I'll present here is very much I would call scientific machine learning or machine learning for science. We really try to bring in science into machine learning algorithms. At the same time, we want to use machine learning to push forward science development, and we interact between the two. And we have high standards for this type of algorithms. It's not just for some data sets. You really want to make sure it's part of the knowledge of science. And I think the theory of scientific machine learning should really couple itself with insights for practice. And we should also have very good software for other people to use. So my group's approach to scientific machine learning, I'll call integrated. We really have students sit in the wet lab and become as scientists as possible, as much as you can learn on your own and from collaborators. And then we develop machine learning algorithms to solve a particular problem before we generalize to another problem. So we see that in the examples I will cover today. And then we kind of, when we see the empirical evidence, we take a step back and we do try to understand from a theoretical point of view. And interpretation is very important for scientific machine learning, right? You know that we have this EU data protection regulation, give everybody a right to explanation. But in scientific machine learning, it's very important. Most of the science books you open are not made of equations, are made of narratives. So you really have to turn whatever insight from the algorithm, the theory into narrative. And we had this paper with my current and former students to define interpret machine learning, because there were a lot of papers. And so what do we mean by interpretation? So we developed this 3-component framework we call PDR. And the important thing we try to bring out is relevance. The interpretation, you need to have a defined audience and also a problem. So even if it's the same audience, depends on different problem, you might want to come up with different interpretation. You want to use predictive accuracy to really check reality, and you also have descriptive accuracy to communicate. And you can use global predictive accuracy. We can look at a lot of investigate different cases, different data points, or different images, see how the predictions run, not just average. And descriptive accuracy has a lot to do with the audience. And can you really capture the relationship learned by the machine learning models? And the relevancy, I think, is at the heart. And people usually think this way, that you have decision trees is most interpretable. Logistic regression is kind of a little more, less interpretable, but still pretty good. And then you end up with deep learning, which become pretty much black box. So what you will see later, we're going to turn this deep learning in scientific problems into something like wavelets, which is very interpretable in physical sciences. And the relevancy in scientific machine learning really means that the insight have to correspond to the facts. And maybe you can do experiments afterwards to really check some of the facts you learned or the interpretation you learned from a machine learning model. So this is just like an overview. So for the rest of the talk, I'm going to share some results we're very excited about. Just got into NERSP. Oh, it's going to happen, I think, this week or next week. Adaptive wave distillation from like ResNet, like 18 layers to something like 30 coefficients. And we started with a cosmology problem, and then we just used the same methodology as a generalization for a cellular biology problem, and also gave very good results. That's kind of supervised learning. And then I will turn to unsupervised learning, because wavelets can also be learned with unlabeled data from natural images. And we have been working with these Drosophila biologists and also neuroscientists from Allen Institute, just use unsupervised learning. And you don't get wavelets. Depends on what's input. The dictionary learning will actually give you other biological meaningful partitions that are not wavelets at all. So this really brings in the importance of the input distribution to these machine learning algorithms. And now I'll end with some local and global identifiability and theoretical results on dictionary learning. So the first kind of motivating project was with cosmologists from, at the time, Francois was in Berkeley, now back in France, and my postdoc, current post and current student, Woo-Suk and Chandon, and then Francois' collaborators. So here they have been using deep learning, try to understand some fundamental parameters of cosmology on simulated data. So they have this fundamental parameter, something called omega m. It's about the beginning mass of the universe. And they do forward simulation, and the purple image on the right is from the simulation that's supposed to be the map of mass in the universe. And can you recover, because you know omega m, using machine learning methods? It turns out that convolutional network works really well. The thing is, what does it really learn? What do we really understand how this deep learning do this inversion, right? This is forward simulation, and you kind of end up with this parameter. And then when we talked to them, they said frequency domain is very important. So we want the interpretation in the frequency domain. So you can turn whatever the interpretation you have through this Fourier and inverse Fourier transform and make F prime, and then just now get understanding in the F prime. So a lot of interpretation method can be done through this transformation. And we have a particular one we have been using for natural language problems and also for image problems. So this was developed not for scientific purposes. This was just a generic machine learning interpretation to go beyond just a single feature to get importance for combinations of features. So for this, we could now really give importance, not just for a word, but also combination words. You can have, what's the importance of very good, not just not, very, and good. And you decompose them. It's kind of an calculation approach. And you collect the different terms, let it run through the deep learning network, and then you end up with this importance for single words and also combination words. You can do the same thing for images. Similarly, you can get importance. So when you use this, generalize this to the frequency domain, and now we can give an importance score for each frequency domain, 0.1, 0.2, 0.3, and you end up seeing that there's a peak at 0.3, really saying that the frequency 0.3 is very important for this prediction. And what you have is saying that if you want to do simulation, you want to pay attention to frequency 0.3, but other high frequencies are also important if you want to use deep learning to get omega m. And this is, yes. Yes.
You have some sort of there is some fundamental parameter method, right, that you want to predict.
Yeah, you want to estimate.
But it's not known, right? So how can this? Or maybe
no, this is simulation. So you know that. That's why you can develop a supervised learning method. So they have certain setup, have certain. They also change other parameters. So that's why it's known. So you use this simulation to see whether your method works, and hopefully you use that to estimate the real data. Yeah, in general, it's not known, right? But in simulation, it is. So it's kind of like a sandbox for doing the real estimation. So this is just very toy example to show that if you use other methods like integrate gradient or sharp and deep lift, for this very simple problem, our contextual decomposition CD can really figure out the frequency much better. Well, they do pretty well, but you have quite a big lift for this very simple function. So that's where we were before we jumped into wavelets. So when you do Fourier kind of analysis, the next thing you want to do is probably local Fourier analysis, which is wavelets. And people already actually using wavelets to do estimation for omega m. And this is the newest paper, again, led by Wusuk and Chandon and our collaborator, Francois Lanussi, and biologist Uko from Berkeley Advanced Imaging Center, who is the cell biologist. I'll show you the second example. So here is the idea is that we have this ResNet works pretty well. And can we actually make it a simpler model and something meaningful out of it? And there's already this line of work called model distillation. So you start with a complex like deep learning model. They have done quite a bit into a smaller neural network. So you start and you have this called teacher and student. You take the fitted model and generate new data and you fit a simpler form. People have also done it for editing models. So you start from deep learning and you end up with decision trees, you end up with editing models. So you use this bigger model to guide you and to smoother than the data, and you can, in fact, doing interpolation too. You don't have to really stay with, most time you do, but you don't have to, where you have the observations. So we want to do, because we in this physics domain, we want to learn wavelet transform instead of doing decision tree or editing models. And the goal is to improve interpretability, compression, and also efficiency. So here's one slide tutorial on wavelets. Time series just tile the time and frequency domain through just bars. You don't worry about frequency, just look at every time point. And the frequency domain, Fourier transform, you look at only frequency domain, you ignore the time. And wavelets, you tile the time and frequency domain in a more strategic manner, so you cover both, but also being local. You have much less in the high frequency, you have much bigger like gap, so you don't pay as much attention to high frequencies, but you have this interaction in time and domain. Time and frequency. So on the left is the Gabor wavelet filters. And if you look at the first layer, AlexNet, you also miscolored. So if you try to filter out the color, you see Gabor-like thing also come up. So Gabor wavelet is kind of mathematization of this observed wavelets like things from supervised learning. You can also get things, just take patches and do unsupervised learning. And there's also neuroscience work in physiology to show that V1 neurons in the primary cortex also care about orientation, location, and frequency. So you also have neuroscience evidence might be real. So what we want to do is we have already a pre-trained DNN from ResNet, which works pretty well for the fundamental parameter omega m of cosmology. And we want to find adaptive wavelets, mother wavelet, that will really make things interpretable. So we end up designing this loss function with three terms. One is reconstruction loss. So you use the wavelet transform. If you use orthogonal wavelet, it's invertible, the phi. And then you put it in the transform domain. And then you put into the pre-trained ResNet. And you want to make sure the XX hat are close. So more or less the phi is invertible. And then we also design a wavelet loss to make sure that the transform have a high pass and low pass filter. And we want to make sure they kind of like wavelets. And then we have a third interpretation loss. So this is taking the ResNet fitted value, and we didn't do our CD. We actually did the gradient, and just it's easier to calculate with PyTorch as a one norm of the gradient, one sparse to be the interpretation. So I have three terms. So let me unpack a bit the second loss. You look at any wavelets book, all these high and low pass filters have to satisfy these equations to be, you start with a mother wavelet and you expand it to these bank filters, and they have to have this equation satisfied. We didn't really deal with 7.31. That's hard to bring into machine learning algorithms, but the other equations, you just turn them into constraints. So that's part of the wavelet loss. And you have more constraints, we just bring in, that's for the low pass filter. And both necessary and sufficient conditions, we brought it in as equation constraints. And there are also sufficient necessary conditions for high pass filters, and we penalize as well. Yes. Yeah.
Sufficient conditions for what?
This condition for the high pass and low pass filters. So you start with of a mother wavelet, and then you can make up these high pass and low pass filters. You have translation and scaling just automatically made in the wavelet kind of enterprise. You have a certain way of moving it around and shrink it, and that give you the high pass and low pass filters. So for any mother wavelet, you can just design the high pass, low pass in a very systematic way.
So then mathematical definition of I thought high pass is just the one which passes high frequency. There is some type of mathematical definition.
Yeah, you can just that's the G, right? That's the low, that's the high pass, the G, right? So you have the mother wavelet, it's called a scaling function. And you have a conjugate Miller filter, H, that's the low pass. And then you have all this in the Fourier transform, you can just write them down. So giving a mother wavelet, or they call a scaling function, you can just write this everything down. And this is a condition on these high pass and low pass filters. So we just turned off this. So we explicitly looking for the G and H's. And we turn this all into constraints. So the equations become constraints. That's what you see how people use for solving PDEs with deep learning. And we do fix the number scale. In the old application, we only did 5. So that you make a choice. So it's not exactly inverse transform. And this I already said, you just put these two transform together and you take the partial derivative. That's our kind of interpretation loss function. We took L1 norm of that. If you simulate using our approach, so you have a synthetic input and we took a fully connected net, and you can distill this wavelet. So you start with wavelets, add some noise, and then you go through and you fit the deep learning, and you get the distilled wavelet more or less equal to the ground truth wavelet. So this is a proof of concept. And we did a little stability in the sense that we have perturbed the input. So you can see that if you have Sim5, that's a very well-known wavelet, then the AWD get more or less, and if you add noise, you still recover the kind of Debussy5, and this Kaufman 2. So this is just the one-dimensional case. And this is to show, again, how initialization might change things. There's change, but you still more or less get the wavelets back. So back to the cosmology problem. We already designed, Francois already designed ResNet. And we now have this distilled wavelet. It turns out that the cosmologists already use fixed wavelets to estimate omega m through something called the peak counting algorithm. So what we did is that we learned the distilled wavelets and we plug into the peak counting method already in the literature. And that's the result. So the prediction error, the lower, the better. It's actually 10 to the minus 4. It's pretty very high precision. And the peak height, it means you just use the peak height, just counting without doing any wavelets. You get 1.6. If you use Laplace kind of wavelet, you get 1.3. You use another wavelet, you do 1.2. You do WG5, you don't do as well, and ResNet does much better than all of that. And if you use AWE, you get another 10% improvement. And if you don't use the interpretation loss, saying that how would you get if you just do wavelets without doing the deep learning part? And you don't do as well. So this really shows that deep learning really helps in terms of estimate omega m beyond just getting adaptive wavelets. So the peak counting algorithm is in the literature. You just bring into histograms and then you know the simulation. So you find kind of like a weighted quadratic distance and you find the right one. So that's just plugging in what people are doing already with fixed wavelets. So you look at what we do here, the wavelets for the cosmology, is that there's some trade-off, right? You have attribution penalty, that's the interpretation penalty, and you also have a sparsity penalty. And we use cross-validity to find this is kind of symmetric wavelets. So this is a method we designed for cosmology. And then we're also working with cell biologists on a different problem, which is how do we observe this process? So it's quite amazing that this is a process how the cells, this is now cell biology, how the cells gets the food, that's one way, into the cell. So this horizontal line is the cell membranes. And first, the cell will lined up cargoes outside the cell. And then it creates a little pocket. And then there are scissors called actin to cut it in, and then you unpack. So this is the process, one way that the cell gets its food into a cell, not through a channel. I didn't know about this until I got this project that I thought it was just an old channel, but actually it's really cut it in and embrace it, and then you get the food in. It's a very central process for cell life called the clustering-mediated endocytosis, CME. So to understand this process, you need to know all the different biomolecules, how do they interact? And one problem they called molecule partners is like which molecules work with what. It's a very challenging problem and central. So the clustering can be observed. This is a video. Try to follow where they are. But not every time you see clustering, the cargo gets successfully put into the cell. So you want to know why it's successful. There's another biomolecule. It's more expensive to follow, the green dots. If you see that, you know the process is completed, not about it. So the question posed to us was, how can you predict for something easy to measure to something harder to measure? So we actually have to process a video into a time series. And then we use LSTM. And then we distill into a wavelet. We end up with 30 coefficients to do the prediction. Directly using deep learning on video actually didn't work very well. we need to really understand the video process and all the Gaussian blurring, all of that is turned into a time series. So directly working on video didn't really work. As we know, video can be challenging for deep learning. So we basically borrowed the idea of AWD from cosmology to this problem. And for here, it's R squared. It's you want to be high. It's like a proportion, like a variance explained. Again, if you use wavelets directly on the time series data, you get 0.2. LSTM get better result 0.23. AWD give you 0.26. So it's about 10 to 15% increase in performance. And if you don't use the interpretation loss, again, you don't do as well as using the interpret. Saying that LSTM does work or help in this case as well. So this is kind of the generalization. You already develop adaptive wavelets, but this is just used for another problem. And you can also see you can see the wavelet find is asymmetric. It's not symmetric as the last one, because here we already figured out by looking at all of the curves. It's really when the predictor really peak and fall. That's a good indication the process will be completed. Any questions? So now we transition to unsupervised dictionary learning, right? We use Gabor because science already is local Fourier transform and physics love Fourier transform. And however, the wavelets can also be kind of derived through image patches from AlexNet. And dictionary learning is actually a simple, effective method in representation learning. But you don't always get wavelets. So that's why dictionary learning is very interesting. Otherwise, we'll just use wavelets. So this is an older project. We try to figure out how a Drosophila fruit fly in the beginning as an embryo, the different parts as color by colors, turn into different organs in the adult fruit fly. So this is developed biology, where you want to figure out the pre-organ regions and how they turn into the adult regions. And when you have the data, so this is a collaboration with Erwin Fries and Sue Seneker and Andy experiments and led by my former student, Siqi Wu, and the postdoc, Joseph was also engaged. So this is an older paper. There's a lot of biological images now. I said there's a lot of science images. People can now really figure out how genes interact where and when. And we're looking at the whole Drosophila life cycle. We're looking at like 2 to 3 hours. So at this, you don't see the shape of the organ. So what you see is this particular gene called AAY, where it's expressed. So this is a developed biology where the different genes, you can think of it already, you have a house, you have already an embryo. And they figured out where the different organs will be built. So you can think of as the different workers work together to build different rooms in this house. And the different genes, depends on where they are, they work with different teams. They don't always work together, just like a painter will be everywhere. So a lot of genes we call ubiquitous genes. We don't really use it because it's everywhere. But other genes are more specific in different locations. And we're interested in gene networks, local gene networks. So people already figured out at this stage, there's something called a fate map. They use ablation, they cut different things out and see which part disappears. So can we reconstruct as a proof of concept that fate map to know what they're going to become? And the different colors means the magenta is a new nerve system. So, you know, where would the nerve system develop in the beginning of the embryo? And this is just to show that our gene expression data is high throughput, has already some information. You can see the two stripes, it's a little mismatch. You can put them together, they kind of inhibit each other. So we want to use modern data to discover this fate map. And there's a very famous gap gene network, a Nobel Prize, that we want to reconstruct as kind of a validation. So that's a fruit fly embryo, that's human embryo. So this is very important network for called segmentation. It's basically how our spines develop. And these genes on the circles, the giant here interact with these 3 genes, and this here only interacts crew. This is all German name. That means if you knock out this gene, the embryo become giant. And this is tailless. If you knock out this gene, it's on the tail. The embryo doesn't, the fly won't have tails. So we use this as a proof of concept. So it's huge amounts of work. So this is another set of scientific images. We have to put them on the same ellipse so that you can do machine learning on. It was huge amount of work to get here. And we use Eric Singh's group's software. This is an open data set. And we also downsampled because otherwise things won't match. So huge amounts of pre-processing. And I was working on neuroscience at the time with Julian Mera, Yuval Benjamini, and we want the fate map, and that's like a decomposition. So I said, let's try dictionary learning. We end up using non-trigonometric factorization, but we started with dictionary learning. It's kind of borrowed from neuroscience. Yes.
This ellipsis, what does it represent?
Next question. So these are different genes. Each image is a different gene. And this says that this gene is expressed a lot here and here. And this gene is everywhere.
I see. It sounds like it's actual like embryo.
Yes, actual embryo after we warped it into this shape. It's not all very nice. Actually, the embryo is 3D, right? It has to be the right facing to the microscope. Otherwise, it's not right. So there's a lot of manual annotation. You have to see the little guy had to lie in the right way when the photo was shot. And then we put them into, this is Eric Singh group's software, put them and then make them into like a comparable images because they have different sizes. Thank you for the question. So this is a different genes. So you hope for the co-occurrence, you figure out the different size of like different pre-organs. So we use dictionary learning. So we turned each gene expression just into a vector without coding the neighborhood information. Just, of course, you need to do a fixed way, make it into a long vector. You do dictionary learning, you do matrix factorization, you end up with this, we call principal patterns. They are just the items. And the very nice thing is that even we didn't put any spatial constraints, the principal patterns came up to be continuous in space. These are the pre-organ regions. These are the basic units, like the room floor plan, that different genes worked together. And the biologists recognize that they're very meaningful. And question is how many we should choose. So we use this stability principle. You take random, so we have random starting points of just subsample the data. So for a fixed case, say 20, and you do many runs because you have many different starting points. And the non-linear matrixization condition one on the other is convex, otherwise it's non-convex. And use correlation to match them. And you look at their distance similarities. Right, so you end up with 2 dictionary sets, or we call principal patterns. You find the best match from one set to the other, and you minus that. If it's 1, you get 0, so they have perfect match. And this dissimilarity can be used over many runs as an index to select a number of components. So there's a clear minimum at 21, and 22 is pretty close. So we did take a look at 22. 22 is a splitting one of the region into 2. And the biologist said, it's too small. So we stayed with 21. So this is using stability to select the number of principal patterns or the number of components. Here they are. I was really amazed that it's almost like you have vertical, horizontal, and anterior and posterior. Very much organized and aligned. And the biologists recognize they're meaningful. But we didn't stop there, just say you said so. We really tried to figure out for different regions, which genes expressed. Now we have this concept of regions, like rooms. And we look at which genes are overexpressed, whether it match with what people know already. And we also did a prediction. We can reconstruct the gap gene network. We miss one link. And then we end up doing some theory. So that's where we start doing theory. And we also did knockout experiments. So the knockout experiments, I would say that we have 40 of them. We still couldn't analyze them because we had a problem finding a phenotype. We found a very important gene was lethal, but still couldn't come up with a story why it's lethal. So you knock out this gene, which had very little information on, the embryo dies at 16th hour. So it's very important. But our local gene network was not right using correlation. So if you lay out all these 21 principal patterns on top of the fate map, now you have conjectured functionality for these regions. Like the one in there is magenta because for this is all schematic. That PP5 probably is about brain development. And you have heme guide that's become the stomach, and you have mesoderm that's about the skin. So all of that, we can conjecture the functionality, and we can see which genes are expressed more here. And then we can actually compare with the literature. So we were able to reconstruct this famous gap gene network except for one link. There was a little bit of evidence, but we couldn't really, our cutoff, which is before we check this, didn't meet our cutoff. So this is kind of a validation that. So we really need to trust these regions we found. And I was very interested in doing some theory on dictionary learning to see when we actually can trust, what are the conditions that will make the dictionary learning work. We couldn't analyze non-incremental factorization, so we went for the easy mathematical tractable bit. There was this work, Gribonwall and Schnaas, and we're looking at actually under, not over complete, but under complete case, and you can fill it in to make it a square case. So Siqi Wu was the lead on the science project. He's also mathematically very able. And he really did all the heavy lifting on the proofs that reformulate the problem because the square case through the matrix inversion into L1 minimization problem. Instead of, you have two parameters, the coefficients and D you want to find, but because the square case, we can turn the alpha problem into a D problem, so we only have one unknown, which is a reference, we call reference dictionary to find. So the literature have this Gaussian, Bernoulli Gaussian model. This is a noiseless case. We assume that, or people assume that you have reference distribution D0 and alpha 1, you have a flip, a Bernoulli flip, and then you have a magnitude of a continuous case, which is Gaussian. So that's called the Bernoulli Gaussian model. With probability P, which controls sparsity, you have keep them or don't keep them. You also have a fixed case when you don't put distribution on alpha, but result pretty similar, so I won't present here. So P is the sparsity. If you look at the, now we have only one unknown, which is D. If you can understand the population version, the sample between the sample and the population can be controlled by the usual complexity theory or uniform convergence. So we concentrate on the population loss function. The first question is when the dictionary will be identifiable. Because now the convex problem, And you can see that the minimum will be sign and permutation invariant, because there's no reason why a particular column is 1 or 2, right? So it's permutation and sign invariant, because you change the sign of the column of D and also change the sign of alpha, you get the same observed data back. The first question is, when the reference distribution, if you have a generative model, will be a local minimum of the population loss function. So here are the prior results that the 2010 paper is a complete case and give a sufficient condition we're comparing a special case for local identifiability. And people also move on to over complete case. We stayed with the complete case. And there was some work on identifiability for non-negative metric factorization. And there are many other works which listed here. So local identifiability, if a dictionary is a local minimum, it's a sign and sign permutation matrix is also a local minimum, because the whole problem is invariant. The local identifiability people usually define through local, through global. The global identifiability usually defines through global minimum, but we're going to introduce another concept to identify the reference distribution, which doesn't depend on global minimum. So we have 2 papers, and the first one was Siqi, as I mentioned. We were able to give basically a necessary and sufficient condition with the equal sign we don't know for local identifiability for the Bernoulli Gaussian model or the K-sparse model. And for the newer work, we're able to remove the Gaussian assumption. So remember, there's a Bernoulli you toss, you remove, and we only need a continuous density for the size. The reason we need that is because we moved from dictionary learning to non-negative metric factorization, that everything's positive for the coefficients. So we want to see whether that will make things easier for ourselves, because empirically we like that better. We start with dictionary learning, we end up imposing a positivity. So this newer work, actually mathematically it's a different approach, it's really Hughes' work, allows us to do the positive, which the early work couldn't. And then we went for some version of global identifiability. It doesn't depend on global minimum. You can construct a case, in this case, that counterexample, the reference distribution is not global minimum, but it has a characterizing property called the sharp local minimum. So to do, to really write out our identifiability conditions, we define a semi-norm based on the random coefficients. So for any matrix A, the 3-bar semi-norm is defined through this formula. Alpha is a random variable, remember the coefficients. And it can be 0 set by with Bernoulli probability P. And it's really can be written in 2 terms. So we can have this almost necessary and sufficient condition for local identifiable, we call Bernoulli type, when you remove the Gaussian assumption. The M is the correlation between the columns of your dictionary, your reference dictionary. And then this matrix you care about, you're going to go through this alpha norm, is that it concerns itself the coefficients and the sign. So this is really about the kind of correlation between the different components of alpha. If it's independent, then if this means 0, this term disappears. This is really related to coherence or incoherence. And this one is kind of a scaling factor on top of the coherence. So if this quantity in the diurnal is less than 1, then the reference distribution is a local minimum of the population. If it's larger than 1, it's not a local minimum. We don't know why it's equal. That's the almost necessary sufficient. It's kind of reminiscent with the irrepresentable condition for the SU, right? You have the mix of the coefficients with signs. It's not the same. And you have some coherence in there, but it's a different form. Yeah.
So it involves the same alphas.
Yeah.
So what does that mean?
It's easier to interpret. It's in this form. It's this matrix is easier to interpret. The norm, I don't think I have a good explanation. In the particular case, you can see it, but otherwise very convoluted. But the matrix, you can see this is about coherence scaled by the size of the coefficient. And this is about the correlation between the different components of the coefficients. Here, you don't even, under the independence assumption, then if this is mean zero, then you have mean zero. Otherwise, they all kind of interact. Some have two correlation terms. One is about dictionary, one is about the coefficient. So the proof is actually like with matrix rewrite, you can get something like a Taylor expansion for the population loss. Not exactly, but you can kind of think that way, and that's how you have kind of Taylor expansion type of thinking going and get the results. So let's look at a simple case, then you can see something. So if you have a reference distribution that the columns has a constant correlation mu, then if you have a sparse Gaussian with sparsity S, K times P, because the result very equivalent, then you have the nice trade-off, right? When the sparsity is high, you need the coherence to be low. If you have very low sparsity, you have very few coefficients, non-zero, then you can tolerate them all dependence. So that's kind of the bound. It's not 1 over S, but like 1 over square root of S. But the interesting bit is if you have a non-negative Gaussian, because remember we want to go to non-negativity, because we end up using non-negative factorization. And you have kind of a weird thing going on here, is that when you have high, like very dense, and mu is high, it kind of helps instead of hurts. That's the opposite of what I would think. And one possible interpretation we came out with is that when you have mu very high in this case, because positivity, you have much smaller search space somehow, and that makes things easier for yourself. So the positivity has a very funny interaction with the sparsity and incoherence that I wouldn't have imagined unless you have all these derivations and have the results out. And this is, again, for that simple, like a coherence, symmetric coherence matrix. We basically got the yellow curve. And this is the bounds from previous, people have got upper bound, got lower bound, and we basically characterize the whole boundary for when things work, when things doesn't work.
Just on the previous slide, so to derive those conditions on the interference even, calculating the semi-norm.
Yeah.
Okay, so you can like exactly.
Yeah. So that the 2D example is even easier, right? So the 2D example, you can just get exactly the relationship, right? For Gaussian without the negative and the positivity constraint, you need the coherence to be less than 1 minus P. But if you have non-negative, you don't need any condition on sparsity. Any coherence would be fine. And same example, you can just plug in and you can plot and see that the condition really tells you when you will get the reference distribution will be the population minimum. So for global identifiability, people just define through global minimum. And there are various conditions about, you have constraints on the sparsity, and the Arara paper start with some clustering, and then they do negative coefficients. And then here's a counterexample that for this example, you can see that the global minimum is not, the reference distribution is not, the generating reference distribution is not the global minimum. So the Z has a mixture of Gaussian, that's the non-zero part. And then you multiply by a Bernoulli, which is Cassie, with probability P being non-zero. Because you're projecting for every component, you end up having the alpha has 8 arms, not 4. Because you set something here, one being 0, get projected there and get projected. So you end up with this 8 kind of arm distribution for alpha. And you can show that the reference distribution are generated is actually not a global minimum, and the global minimum is achieved by something related to the green. So this is the case that if you want to do this problem, you cannot stay with global minimum. So we end up borrowing this old concept, well, old, at least from the 70s, called the sharp global minimum from optimization defined in that way. So you kind of lower bounding with the Frobenius norm, just a little bit padding and not the square. Then it's called the sharp local minimum. And then we can show that when this condition holds, that we see for local minimum, it's a unique sharp local minimum. So you don't look at, it's a global property because no other dictionary will satisfy that. And this is uniquely defined the reference dictionary. It's the only one that holds, satisfy the sharp local minimum. But you cannot stay with the global minimum. Here's a very quick idea on the proof. Since we can do the inverse, and you basically show that this vector is exactly k-1 in the subspace of k-1. And from this, you can kind of prove that other dictionaries don't satisfy that, and therefore they cannot be sharp global minimum. And how do you check the sharp global minimum? We designed a kind of stability type of numerical algorithm. And it's kind of reminiscent to the stability for selecting the number of components as it is. It's not exactly the same, but it's like, if things perturb, things still doesn't change much, then you know it's a sharp global minimum. And more recently, we've been using this dictionary learning for another genomics data from the Allen Institute for Brain Science to collaborate with my former student Riza, now UCSF, and people and also Hugh Wang, who was on the lead also on the theory project, and a bunch of people from Allen Institute. This is a 3D gene expression for adult mouse brain. And people also hand annotated the different brain regions, but this will make it data-driven. So what they like about what we came up with, there's some parts they already know. Other parts, we have more refined regions than they knew, smaller. The big one, they kind of knew, so that's why we end up with 18 principal patterns here instead of, so there was a local minimum at 13. And through discussions, we still took the global minimum at 18 because they have more regions than what people knew already. To summarize, I was really advocating for more people get interested in scientific machine learning, and interpretation is hugely important for scientific machine learning. And we use science to derive the adaptive wavelet, which worked for cosmology. That was a motivating case. And then we just borrowed it for another problem, and that worked really well. And then dictionary learning, we have been very successful using it. Oh, it's variant, non-geometric factorization. They're not the same as, you know, the positivity for two biological problems, and both gene expression, one in neuroscience, one in developmental biology. And we're able to do some theory kind of supporting the use of non-negativity, because with that constraint, you have more relaxed conditions. So the current directions, right? So we went for wavelets for cosmology without doing dictionary learning. Because we like wavelets, people use wavelets and make sense there, and we use it. But if you use dictionary learning on these cosmology images, if you take a small 12 by 12 patches, you don't get any pattern out. If you take the whole image, use independent component analysis, you at least get something out. So definitely there's very strong inductive biases. Depends on the formulation, depends on the algorithm. And we need more investigation. Just because these two were separate projects, just this week I get Woo-Suk to help me to get some of this. The question is, do we find new patterns? Are they meaningful? Are we adding anything to the wavelets that people already doing? And for the both theoretical front, right, there's a lot of, we have actually in that paper, iterary algorithm, but not really practical. It's like for we specify models, then it just doesn't work. And there are many good works on local convergence, including one from Peter and Aurora's group, but it's local convergence. And there's a very interesting paper from 2010 from Banju's group. So autoencoder is kind of another version of dictionary learning multi-layers, and you add noise to it. If you don't add noise to it, autoencoder wouldn't give you wavelets. But if you add noise to it, it gives you autoencoder. Can we show some local convergence of this very interesting algorithm? So that's where we are. And to leave you, like, I started with ImageNet, but a lot of very exciting scientific image set that hopefully will help to further develop deep learning and science. Thank you.
Maybe one question, and then we can go to the.
Yeah. So the notion of interpretability that you use with the deep learning, as part of the pipeline is a sort of sparsity of this.
Yeah, adaptive wavelet.
Yeah.
Yeah.
So, was that? I guess there could be many other.
Yes. Yes. So, for example, we quite like our CD, but it's harder to program, and because you do derivative PyTorch, just make the programming very easy.
I guess one advantage there is you have this very non-interpretable thing.
Yeah.
But it's surrounded by something that, is in some sense simple.
Yeah, I think there's something fundamental. I think wavelets or free domain. I think my conjecture is that there's certain class of problems that relate to physical science. Just one layer of wavelets is kind of enough for those problems. But somehow the deep learning still helped, which I don't quite understand. Somehow I went into a space that if you just directly do adaptive wavelets without the interpretation part, you get worse results. I don't quite fully understand, but somehow that seems to be there's a value to going to deep learning. And there will be also some cases where wavelets, so when we try to add layers of wavelets, it doesn't work, because I think after the vision, at least for vision, after the first layer, the other layers, you need other kind of filters. In hindsight, it was pretty obvious, but we did try. So yeah, it's L1. would love to program for CD, but just like the programming is just too much. At least we don't have a package to use, right? Everything we coded up and taking, yeah. So that's why we went for L1. We thought that we'll get worse prediction performance. We're happy just to go for something more concise. Like for the, we end up with like just 30 like coefficients or something, right? With rest that have huge number relatively, but the prediction also got better. That was a nice surprise. There's a lot of images I think out there now. I think the technology of imaging just really advancing.
Good, thank you again.
Thank you.
I think we'll have the next session right at the.
Yeah, that's right.